So I'm going to start off just to warn you, this is, as psychologists, we know there's good stress and bad stress, and this is only about the worst stress. So this is a very dark story, but there is some light at the end of the tunnel. We do talk about some positive ways to moderate stress at the end. So I'm going to start off with a quote from really one of the fathers of stress science, Hans Selye. And he said, "Every stress leaves an indelible scar, and the organism pays for its survival after a stressful situation by becoming a little bit older." So where is that scar? Where is the hidden in the body, this marker of age? And why do we even care? We can just see when people get disease. If we have a marker of biological age that's independent of disease, we can use this early on in life as a developmental marker and learn more about our health, behaviors, emotions, and thoughts, and how to modulate those. So this marker could be really helpful for learning about our own mind-body connection. So my story actually hinges on a discovery that was about 25 years ago by my colleague, Elizabeth Blackburn. She is a molecular biologist who wanted to understand how the chromosomes protected themselves. There's these packages of base pairs at the end of the chromosomes, the telomeres, that protected them, but they grew and they shrunk, and there must be some factor controlling them and modulating them. So she and her graduate student started studying the tetrahymena, and they discovered the enzyme telomerase. And when they upregulated telomerase, this cell became immortalized. When they knocked it down, the cell died. Does this have any relevance for human health 25 years later? This marker turns out to be one of the strongest predictors of early diseases of aging, and in many studies, early mortality. So it not only predicts the life of the cell, but it also predicts the health span of humans when they get diseases of aging. So here you can see the telomere, and these are the just repeat base pairs, TTAGG. They don't code for genes, they just protect. They're very sensitive to the intracellular environment. And as the cell divides, these shorten, the telomeres shorten, and when they become too short, the cell sends DNA damage signals, and this can be a catastrophic event for the cell. So to prevent this shortening with cell replication, thank goodness there's this telomerase enzyme. And this is a really remarkable enzyme. It has a protein component and an RNA reverse transcriptase component that actually rebuilds the base pairs. So for those of you who like to see inside and really understand what that looks like, here it is. This is the telomere length. There's a short end, and there's some mechanical problems. The DNA polymerase cannot fully replicate the short end of the telomere. So that's why as the cell divides, the telomere gets shorter and shorter. Telomerase, because of this reverse transcriptase, actually rebuilds back to the base pairs. So what happens when we don't have enough telomerase? We now know from very recent research that this intracellular machinery goes awry. So when there are DNA damage signals from the genome, from these short telomeres, this both leads to apoptosis or senescence of the cell, or in addition, it's signaling the mitochondria, the energy stores of the cell, that there's trouble. The mitochondria become very dysregulated, the cell doesn't have enough energy or ATP, and the mitochondria start releasing reactive oxygen species or free radicals. So this is a cellular state of extreme stress and probably the basis of cellular aging. So I'm going to be talking about telomere length today, but I can tell you that we are now studying the mitochondria system in all of our studies, and these two are very close friends, and much of what I will tell you is also reflected in the mitochondria. So short telomeres are both associated with chronic diseases of aging and predictive of chronic diseases of aging. So about 10 years ago, we asked whether chronic psychological stress might be wearing down this telomerase early, which would lead to shorter telomeres. So in our first, so I'm going to talk about what we've learned in the past 10 years and tell you, just to summarize, that I'm going to present a lifespan model, because when we look cross-sectionally at people, we're just getting a glimpse of what's really happening, and we have to understand their early development to really understand how they ended up with short telomeres. And after that, I'm going to go into our current research and give you a more granular view. So of course, we want more specificity to understand this stress-cell aging relationship. So one question is, do all cells shorten equally, or is there kind of a driver of cell aging? So we've gone deep into the immune system, and we have an answer for you today. And then the second piece is, what does it mean? What is chronic stress, and how does that play out on a daily basis? What are chronic stress, or being a caregiver is just the category? What are the actual contents? And if we can identify how chronic stress plays out, we can actually target these stress processes, these cognitive emotional stress processes that play out on a daily basis. So back to our first study, this was our hypothesis. We examined a group of healthy young women, premenopausal women, who were either healthy controls or caregiving for a child with a chronic condition. And in that first study, what we saw was that there wasn't a group difference, and there was a difference in perceived stress. This is a classic principle in stress research. It's not exposure as much as it is response. So the women with the highest stress levels had 550 base pairs missing, in a sense, from their telomeres. And given how many base pairs we lose each year, this was a whopping effect size, and roughly what you would lose in about 10 years. In terms of their telomerase, they had about 40% lower levels of telomerase activity. Now, you could say, well, perceived stress, that's personality and neuroticism. This is just a correlation. Genetics might explain it. But we had another stress measure that was more objective, and that's exposure. How many years had these women been caregiving? And here you can see that the more years of caregiving, the shorter the telomere length. So let's fast forward 10 years. And I'll just ask you, do our telomeres really care what's on our mind if we're anxious, depressed, ruminating, chronically stressed? There have been numerous studies looking at different aspects of stress and trauma, and there have been an emerging consistent literature with psychiatric disorders of emotion regulation as well, particularly major depression. And there's also a dose-response relationship in these studies as well with duration. So I will admit that my colleagues and I have done probably a lot of maybe a large percentage of these telomere stress studies over the last 10 years, trying to hammer out and understand how behavior and psychological factors are affected by, are affecting this cell aging system. And I am the first to admit that that I can be called boring for sticking with this same nature of this relationship for so many years. But I'm mentioning it because I feel like this group would appreciate that we have a highly replicable finding. So when does this start? How early can this relationship start? We think of this as a wear and tear process and a marker of aging. That's how we thought of it 10 years ago. We think of it very differently now. This field started with several studies where people looked at early trauma and just asked adults their experience of early childhood adversity. And these studies have shown that there is this dose-response relationship. Here's a study by Jan Kiekel-Glaser. People with no early adversities had the longest telomeres, and people with multiple early childhood adversities had the shortest telomeres. So that leads to the question, is this a retrospective recall bias, or is this really happening during childhood? So in the last 3 years, there have been 6 studies that have looked at children prospectively as well and found that bullying, domestic violence, social impoverishment, abuse, neglect, all of these are associated with telomere shortening online each year in children. And this has led researchers to be very interested in this measure because it may serve as a deep biomarker of early life stress. And if we have such a biomarker, that can help us have a barometer. We can understand damage done, future vulnerability, and possibly even the efficacy of interventions and how those might repair this cell aging system. The Reversibility Network initiative at NIA. So let's go back even further. Does it start in early childhood, or could it even start in the womb? With our colleagues at Irvine, Sonia Entringer and Patik Wadhwa, we've started to assess pregnant women. And in Sonia's first study, she asked healthy young adults to assess and interview their mothers about their major life events, loss, death, et cetera, during their pregnancy. And here's what she found. The adults who were exposed to prenatal stress had significantly shorter telomeres. And in her next study, we've actually gone to pregnant women and directly assessed anxiety, pregnancy anxiety, and that was associated with shorter telomere length in the cord blood. So this is shocking. To me, this has tremendous implications for public health about where we need to start our efforts when we think about aging and health. So I'm going to put that aside. If you can forget about that and not think about your own scars. This is our model. This is with Eli Putterman, where rather than, we think of cholesterol and these risk factors for health as accumulating with age. And when we get over 50, we're more at risk. We need to start monitoring them. So that's how we thought of telomere length. But actually, it really doesn't track that way. Most of the action is in early life, so prenatal and early childhood. These are what's showing up in adulthood. Yesterday, Eli Putterman presented one of the largest telomere studies of 5,000 people in the health and retirement study, and showed that cumulative life stress predicted shorter telomeres in these older adults. But when you broke it down, what was really the significant driver? Early childhood stress. Okay, so let's try to put that aside and now think about mechanism. How can we better understand this relationship in the immune system? Some people think of telomere shortening in immune cells, meaning all the immune cells are shortened equally in all the cells in the body. And while there is some correlation between telomere length across our cells, there is specificity. So we got to look under the lamppost here. Immunologists have been years ahead of us in finding out how aging, basic chronological aging, shapes the immune system. So we learned from them, and let me tell you what we've learned, that among all these cells in the immune system and among the lymphocytes, there is a cell that is responsible for early senescence of the immune system. And that is the CD8 or cytotoxic cell. And here's what happens. As we chronologically age, this cell loses this really important marker, the CD28, and it can no longer turn over and proliferate and recognize antigens and attack infected cells. So we get a weaker immune system. Sheldon Cohen published a paper in JAMA last year where he exposed young, healthy adults to the rhinovirus, put them in quarantine, and the predictor of getting a cold was not overall telomere length, but was this CD8 28 minus cells. That means these cells have lost their 28 minus. And so now we know even in young people, this early immune senescence is predicting important health outcomes. So here's the question. Sorry, this is just another demonstration that older people have an accumulation of these pre-senescent cells. What's so bad about them? Not only are they not doing their job fighting any infectious agents, they are causing mischief. They actually become a little machine for secreting pro-inflammatory cytokines, one of the major pathways of aging. So the fewer of these, the better. To address this question about chronic stress, in our current study, we are looking at parents of children with autism and parents of healthy neurotypical children. So let me go right to the main question. Does chronic stress mimic chronological aging, leading to early replicative senescence of these CD8 28 minus cells? And I forgot to tell you, the cause of losing this 28 minus is low telomerase. When these cells replicate and over time they get low telomerase, short telomeres, and that low telomerase predicts this surface marker falling off. So it's so all we need to do is sort cells, and it's not a small task. There's a whole room that has a machine that fills a small room where we sort these cells, and we pay a lot. But we needed to address this question of where the defect was. So we've sorted cells, and we've looked inside the CD8 28 minus cells. We've measured their telomerase and their telomere length. And then we followed these parents over a year and a half. So what did we find? When we compare the caregivers to the controls and the different lymphocytes, there's no difference in telomerase in their B cells, their CD4 cells, the CD8 28 plus cells, but their CD8 28 minus cells have significantly lower telomerase. Bad news. So what do we predict that's going to lead to? Over time, shorter telomeres and senescent CD8 cells. So here's the telomere length finding. The telomere length is similar across the lymphocytes in these healthy young women, except for in the CD8 28- cells. They were actually similar at baseline. They were a bit short in the caregivers. But over a year and a half, this is a change score, the caregivers lost more base pairs. Their telomeres shortened in an accelerated way in these CD8 20- cells. So we're seeing that just being a caregiver, just knowing that about someone, is predicting greater acceleration of immune senescence. So now let me turn to this more granular question about how can we understand why these caregivers on a daily basis are having this premature aging. So we know that there's biological embedding from early life. We know that caregiving is associated with cell aging as well. But how do these shape daily experience? What are the persistent risk mechanisms that we carry with us every day that shape our thoughts and feelings as we encounter different stressors? So I'm going to admit that my colleagues and I are a little bit obsessed with threat and challenge. And we like to expose people to acute stressors and measure their pre-appraisal. Before they're even having to give a speech and do math tasks in front of these cold-faced, stony-faced evaluators, we ask them how they think they're going to do. And so this is Wendy Mendez's scale for threat and challenge. You can see that there are really different emotional profiles of thoughts that go along with threat. Worry, anxiety, uncertainty, feeling like you can't cope well, versus that positive challenge state. There's even emoticons for threat and challenge. So we know that threat is associated with a bunch of, different neural sequelae in the brain. We just heard that little mice, when they see the predator, it's that initial response they have, the threat response, in their prefrontal cortex amygdala circuit that is predicting their behavioral response later. And so what do we know in people? Just asking people this thin slice question of assessing level of threat and challenge before they're even encountering the stressor is predicting not just brain response, but all these peripheral outputs of reactivity and lastly, this biomarker of early aging, shorter telomere length. So this is what we're interested in assessing in daily life. When people first wake up, we know their cortisol shoots up. What are they thinking? How is the life of the caregiver different? So we wanted to assess waking up in the daily stressor, and so we went in and assessed feelings of control and happiness, worry, unpredictability. Caregivers were higher in the negative appraisals. So guess which of these are going to be most associated with the low telomerase? Unpredictability, that was one of our guesses. We were wrong. Telomerase was higher for the positive appraisals of looking forward to the day and feeling happy, and lower with the stress appraisals. In terms of threat appraisals to their most severe stressor, telomerase in the CD8 28- cells specifically, again, in all of these slides I'm showing you, was lower with greater threat appraisals of their daily stressor and greater anger responses. So boring, boring, boring. I keep showing you that caregiving and other stressors are associated with lower telomerase and shorter telomere length. So I've just told you it's very specific. If you're looking at young, healthy people, we can see this defect solely in the CD8 cells. And now I'm telling you that we're also finding mediation from daily experience. Waking up is important, how people are appraising their stressors. These are giving us targets for intervention. And that's why we're excited about understanding the cognitive emotional online responses. And now we're measuring them on a daily basis. So of course we've been interested in mindfulness. Mindfulness training can help with self-regulation of emotion and appraisal. About 6 years ago, we hypothesized before we had any data that mindfulness would be changing these threat appraisals and damping down the stress pathway and increasing the positive physiology pathway. So Wendy Mendez and Eli Putterman, we wanted to examine, are people who in a trait-like way are more present and engaged in their life versus mind-wandering, do they have longer telomeres? And some of you might know this study, the iPhone study, where they found that over time, people who did more unpleasant mind wandering were much less happy. So we used those items and we adapted them to a trait style. And you can see that people who were very high in mind wandering and low in presence had the shortest telomeres. And vice versa, people who were more engaged in their life had longer telomeres. This is controlling for stress and depression, rumination, all the factors we know affect telomere length. So on a trait-like level, it looks like there's a correlation. What if we manipulate this? What about mindfulness interventions? I was thrilled to find that there was a meta-analysis that just came out. I didn't know you could do a meta-analysis with 4 studies, and for full disclosure, 3 of those are our group's collaborative studies. But it does look like there's a signal. These are all early preliminary studies with small sample sizes, and some did not have active control groups. But it is a promising signal that mindfulness training can boost telomerase. We're trying to look at mindfulness with our caregivers, and of course we're using technology to help insert present-oriented mindfulness within the day, not just a menu of short meditations, but pings that say things that help people become aware of their body and emotional state. So I have about 30 seconds left, and I didn't warn you that you might think about your own life during this talk, and what's on your mind, and whether your telomeres have been listening. So I've told you that that there is what I think of as this uncomfortable intimacy between our thought and emotional processes and how much our cells are responding to this, at least with telomerase. Telomeres are being shaped through life, particularly through early adversity. That's a particularly sensitive time that appears to track through life. And you'll hear in the next talk from Tom Boyce that when we think about stress, we shouldn't expect main effects, but rather differential susceptibility, that people come with their own vulnerabilities and that there are interactions throughout a lifespan. And the good news is that these daily cognitive emotional responses are more proximally and strongly associated to this enzyme, telomerase enzyme, and that we think interventions, behavioral interventions, I haven't talked about those, but there is some promise there, and psychological interventions are going to be effective for slowing down the telomere attrition that we're all experiencing. Thank you.